Am I good? Yeah, I'm good to go. So my PhD, which was finished many years ago, we looked at we needed to understand what was happening with the genetics of Southern Ground-hornbills to know what sort of management decisions we needed to be making to make sure that we're taking genetic health and things like inbreeding and things like that into account. And what we found we weren't 100% confident in the results that came out from the thesis. And so we reran all of the analyses again and added multiple more gene regions and markers. And the answer remains the same. And it surprised us, but it's a pretty cool study. And I'm going to try and take you through. If this looks like I've just cut and pasted from a manuscript, it's because we finished the manuscript yesterday. And so it's fresh off the press. But yeah, so this is what we've got and what we need to work with. So I spent a lot of time trying to get enough samples from across the full sub-Saharan range, and we ended up with a total of 360 samples, and that was I was given the go-ahead by the lab that was probably going to be enough. Obviously, there's no ways we could travel all over the range to capture birds and get blood samples, but we were able to get DNA samples from museum specimens. And so we ended up basically raiding the collections around the world for any and all ground-hornbill specimens. These are some in the Windhoek Museum, and all we needed was a little piece of toe pad. And thankfully, on a bird this size, collection managers weren't that traumatized by us taking a little piece off the collections. And so through this, we were able to add a number of smaller range states and parts of the population that we weren't able to do through live collection. We did manage, however, to do live collection throughout South Africa. We went into Mozambique, Namibia, and Zimbabwe. It was a truly lovely time. It was just so good to be out in the field, to be seeing ground-hornbills in all manner of habitats, communities. Yeah, it was a real experience to be able to do this. And because we were trapping, we spent a lot of time watching birds come into the trap, watching them interact with the decoys, with each other, what they did with the food items before they got trapped, group size. Yeah, it was an incredible learning opportunity to spend time with these wild birds and then finally get them in hand. And we had varied success. This is our only sample from Gorongosa National Park in Mozambique. Because of the history of the war there, those birds are so incredibly skittish that although there was one group every 3 kilometers, so, in theory, not short of ground-hornbills at all, they proved almost impossible to capture. So we spent a month in the field, going out every morning at 3 in the morning, and towards the end of the month, we finally were able to get this female in the hands. So she's probably the most expensive DNA sample known to mankind, but also it wasn't harmful to spend time in Gorongosa. So that was also just lovely. And then we found in areas where the birds spend time with people, it was much, much easier to capture them. This is what we call our super group in Ishawi. It's a group of 9, and within 2 minutes of calling them in, we had all 9 in the trap. It was only Natasha and myself, so there was absolutely no way we were able to process that many birds. And this was me going closer and closer and closer, trying to chase some out so we could just get a representative sample from that group. And they also absolutely destroyed our decoy. Pedro is the name of our decoy. And he had to go back to the taxidermist for an awful lot of repair. There were fragments of fiberglass all over the place. So yeah, such varied responses across the range and between groups and between land uses and countries. And so we caught and we caught and we caught until we felt that we had enough samples that we could come home. And then started the bit that absolutely terrifies me, and I'm going to try and take you through this. So this is this includes all the additional testing that we've done since my PhD. So we first genotyped all the samples for polymorphism at 16 species-specific microsats. We had 6 originally that were published by Agarwal. So we developed 10 more using next-gen sequencing and using 200 base pair paired sequences. We then looked at deviations from Hardy-Weinberg. Linkage disequilibrium was tested for non-random associations between alleles of different loci. And I'm not going to go through all of the tests, but basically we did anything and everything that needed to be done to make sure we had the best possible understanding of what was happening both within and between populations. So looking at variation between populations, between samples within a population, and even variation then within an individual sample. And it went on and on. We tested for isolation by distance and you know, looking at genetic relationships within groups. And yeah, anyway, a lot was done, mostly by the brains at the Sandby lab. And then on top of that, we also then did the gene sequencing. So for the mitochondrial DNA analysis, we used a Southern Ground-hornbill as the in group, but then we also added 3 sequences for out groups. So we used the Northern Ground-hornbill, and then Aceros waldeni, and then the Rhinoceros hornbill as well. And then for the nuclear sequence analysis, we used Bucerus bicornis and Anthraceros albirostris also as the out groups. So we used 1 nuclear gene region, G3PDH, and then we used 4 mitochondrial genes. So we used ND2, COI, Site B, and then we also had both 16 and 12S ribosomal RNA genes. And then we did everything that we have to do for that. And I'm not going to bore you with this. Rob told me to just skip to the end and tell you what the byline is. So in terms of results, for the 16 microsat markers, we found 112 alleles across 13 populations, the 13 populations that we sort of clustered them as rather than what the data showed. All loci tested were polymorphic, with the number of alleles being anything from 2 to 14. And we found no null alleles at all in any of our samples. And again, I'm going to just skip through the nitty-gritty bits because I'm getting funny looks on this side. And when we looked at population and gene flow estimation, though, we found that there was a little bit of clustering, but essentially all that happened was the individuals from Angola, Botswana, Kenya, Mozambique, Tanzania, Zambia, Namibia, and DRC clustered together. And so that's what we called the northern part of the range. And then the South African samples, Eastern Cape, Kruger, or the Lowvelds, and KZN all clustered out into what we are calling the southern cluster. But when we then looked at that through the PCA, there was absolute lack of population differentiation. So, you know, that differentiation is really, really tiny. And we found that the variability was more within a population at 76%, whereas amongst groups, it was really tiny at just 6%. So really weak differentiation, and that is across the far extremes of the range. So there was that little bit of evidence for isolation by distance. And in total, we only found 17 private alleles, and that only constituted 15% of all alleles, and that the frequency of these private alleles occurred at really low frequencies. And then again, when we looked at the population assignment, there was weak genetic structure across the range. And so all we can ever do for a ground-hornbill sample is assign it to a northern, basically South Africa and everything else. So if we have a look at the tree, you can see that there's a few tiny little clusters that come out, but and that's when they compared to Buceros and Anthraceros. But when we then put in the full reference sequences into the tree, all the structure totally disappears. And that's because at most there's a 2 nucleotide difference between any of the locations. And so when you look at the PCA, there's absolutely nothing coming out of that. And again, just no sign of subpopulations at all, regardless of which test, regardless of whether we were looking at the gene sequencing or the microsats. And so what does that mean then at the end of the day? So this is the first assessment for ground-hornbills across the full range. It's also the first for a large flighted African savanna bird. And what we found is there's an absence of any significant difference between the expected and observed values. So we have no inbreeding across the entire range, and that South Africa is only a little bit different from the rest. And what does that mean then for conservation, which is ultimately what we need to think about here? Essentially, Southern Ground-hornbill the Southern Ground-hornbill population is pan-mictic. There's a tiny bit of isolation by distance, and that's only at the extremes of the range. So within South Africa, there's absolutely no population differences at all. Unfortunately, what this means, though, is we can't use genetics at all for checking population assignments. So if we wanted to monitor trade, if a bird is confiscated in trade, or individuals are found in a muti market, we would be unable to say where that bird comes from or what population it comes from. And so what we're going to do instead is rather look at relatedness within the traditional medicine markets. Are the birds that are coming into the markets whole groups? As in, are they all related to each other? Or is it just an annual offtake of juveniles? As Kyle said in his presentation, it really depends on who in a group is the important group member, and so obviously this has important repercussions for any harvest for the traditional medicine trade or aviculture, because if we're removing those experienced birds, there's very little chance that the rest of those youngsters are going to be able to learn the skills they need to get into that position themselves. So this is something that we're going to be looking at going forward, and I'll discuss that a little bit more now. So we will continue, as we have been doing, managing the South African and the Tanzanian captive stocks as separate management units, just following precautionary principle. If we were down to the last 1,000 ground-hornbills in the world, then it wouldn't matter. But we don't have to, so we shouldn't. And we have already been managing the stocks as such. The only real shame with that is there are many more Tanzanian or northern birds in the global captive population. So it would have been nice if there wasn't even that difference, because we would have been able to bring in eggs or chicks for rearing for the reintroduction program here. But we're going to continue to manage these stocks as distinct, not distinct, but we'll manage them separately. And so locally within South Africa, there is no differentiation that we could pick up at all. But luckily for things like the restoration program, the reintroductions, a bird can be harvested from a nest in the Eastern Cape and used for reintroduction in the north of the country without us having any genetic risks with what we're doing. So that makes it much clearer for us to go forward with that. But we do also have to think about it, and that came out clearly in Evie's talk, is we must maintain natural gene flow. Just because we're sitting with quite a nice situation at the moment in terms of not having to manage little subpopulations, the more fragmented the Eastern Cape becomes, if we are unable to resolve that gap between Southern Kruger and Northern Zululand, we may start seeing differentiation that in time might lead to those more southern populations not getting the restocking as such that they need. We definitely don't want to end up in a position where we have to do meta-population management. We would much rather try and do everything we can to restock and support populations to maintain natural gene flow. And so, you know, across the range, it supports the hypothesis that they are generally quite habitat generalists. You know, there's certainly nothing within the whole of the sub-Saharan range that has acted as any sort of barrier. And also it fits beautifully with Kate's data on the dispersal distances. So we know that the young females disperse a minimum of 4 territories from home. If not, you know, if not more. That's just within the APNR, where the population is at capacity. And so it's this combination of not being too fussy about habitat, but also the youngsters being able to disperse quite far. That seems to be making sure that this population is doing okay from a genetic point of view. So next, in terms of looking at the molecular health, we do need to try and determine the genetic effect of population size, because, you know, obviously, although we're doing, I think, a fair job and hopefully better over the next 2 years of censusing our local population, there's just no way we can do that at a global size. And the effect of population size is usually an order of magnitude lower than the census figures, because it's lowered by skewed sex ratios, and then also differences in reproductive success amongst individuals. And that, you know, fits exactly what Southern Ground-hornbills do. And this rule of thumb does hold for other savanna cooperative breeders like the African wild dog. The other thing that would be really cool to look at is look at the suture zone, the East African suture zone, where the Southern and Northern Ground-hornbills come together, because this is an area that regularly produces integration and hybridization. So to just be interesting to see what's happening between the 2 sister species up at the top of the range. And then there's a new project at WITS now, a collaboration with SANBI, where they're doing full genome sequencing for both the Northern and Southern Ground-hornbill. So I think hopefully there's going to be a lot more exciting stuff coming out, but it's nice to know that what we weren't sure about, you know, no matter how many more markers and how many more analysis we did, the answer holds. A ground-hornbill in the Eastern Cape is the same as a Zim, is the same as a Kenyan ground-hornbill. And it's really nice to sort of have that nailed down now, and to enable us to go forward with our conservation planning and, more importantly, implementation. Are there any questions on the genetic stuff?